Okay, so this is going to be part 2 of the blood, and I'm going to finish up the formed elements with this particular recording. So the next set of formed elements, we finished talking about erythrocytes, is now going to be leukocytes. And I'm not going to get into too much with the immune function today because once we finish up the cardiovascular system, we're going to get to the immune system. So the leukocytes, these are your white blood cells. As you saw in the blood smear from the first recording, these are much larger in size than the erythrocytes, and they do have a prominent nucleus. So these are actually still fully whole cells. They will use the bloodstream as transportation and generally do not perform their functions within the blood. They will actually leave the blood vessels by adhering to the walls of the blood vessels, squeeze through, and enter the surrounding tissues where they will then take care of their immune functions. Leukocytes are divided into two basic categories: granulocytes, which contain cytoplasmic granules that are going to be released when they're activated, while the agranulocytes will lack these visible granules. And these granules can actually be stained, as you'll see in a second. The granulocytes, these are readily distinguished by their unusual nuclei. It's a single nucleus that is composed of multiple connected lobes, so it actually appears that it has multiple nuclei when in reality it only has one. All cells in this group contain general lysosomal granules as well as granules unique to each type of the granulocyte. And these lysosomal granules will be very important because they allow for digestion of, say, bacteria or whatever they're ingesting. The granulocytes will be divided into the following three categories based on the color the granules appear, either light lilac, dark purple, or red, respectively, when stained with basic, methylene blue, or acidic eosin dye. Neutrophils are the most common leukocyte. They have cytoplasmic granules that will absorb both dyes or stains in their cytoplasm, and it will give a light lilac color. Neutrophils function as active phagocytes that will ingest and destroy bacterial cells. These are usually the first cells that show up to a damaged area and they're your first responders. They're also called polymorphonucleocytes or PMNs. Some of you that might have some clinical experience might be familiar with polys or PMNs. These are your neutrophils. They are uniquely shaped nucleus composed of 3 to 5 lobes. With the neutrophils, what you have is injured cells actually release chemicals. So when you damage tissue, you release chemicals. And by chemotaxis, the neutrophils know that where to exit the bloodstream and then release those granules into the damaged tissue. The granule contents will directly kill the bacterial cells and attract more neutrophils and leukocytes to the region. And this will overall have, as we get to the immune system, what we call an inflammatory response. The eosinophils, these have bilobe nuclei, and they appear red to dark red due to the uptake of the eosin dye. Eosinophils are also phagocytic cells that ingest foreign molecules. They are particularly important for parasitic worms or allergic reactions. And the granules from the eosinophils will contain enzymes and toxins that are specific to parasites. Also, chemicals that mediate inflammation. You'll see most of these are actually going to trigger or mediate an inflammatory response. The basophils, these are much less common. They have an S-shaped nucleus and appear dark purple due to the uptake of the methylene blue dye. And the chemicals in these granules will actually mediate inflammation. So if you have an elevated basophils, usually your inflammatory response is really, really in overdrive. So looking at these, we can see the neutrophils, eosinophils, and basophils. Okay. Now your agranulocytes, these include lymphocytes and monocytes, two very important cell groups. The lymphocytes can be very important in your adaptive immune response with both B cell and T cell pathways. Monocytes will actually mature into macrophages, which are roaming phagocytic cells. So the lymphocytes are the second most common leukocyte in blood. They contain large spherical nuclei and a light blue rim of cytoplasm. Then you have two basic types, similar in appearances, but very different in their functions. Both types are activated by cellular markers found on cells called antigens. We have our B cells or our B lymphocytes. The B cell pathway is called humoral immunity, and this will actually produce antibodies which bind to and remove antigens from tissues. Each population of B lymphocytes secretes antibodies that only bind to a specific unique antigen. Your T lymphocytes or your T cells, these are also activated by specific antigens, but they do not produce antibodies. Instead, they have membrane-bound receptors for individual antigens. T lymphocytes will activate other immune system components and directly destroy abnormal body cells, such as cancer cells or virally infected cells. Antibodies and T cell receptor cells both bind to and only one unique antigen based on the structure. Okay, and again, this is really going back to those structure function core principles. So with the B cell and the T cell activations, these are both going to be part of your adaptive immune response. And again, we'll get to that when we get to the immune system. Monocytes are the largest leukocyte. They actually have a U-shaped nuclei surrounded by a light blue or purple cytoplasm. Monocytes will only circulate in the blood briefly before exiting the capillaries to enter tissues where some will mature into macrophages. Some of you may have heard of macrophages. Macrophages are phagocytic cells that ingest dead and dying cells, bacteria, antigens, and even cell debris. If you damage tissue, you got to clean up that damaged tissue. They will also activate other components of the immune system by displaying phagocytized antigens to other leukocytes. Here we can see a typical lymphocyte, and then here you can see a typical monocyte or macrophage with that U-shaped nucleus there. Now, a lot of times physicians will order what they call a complete white blood cell count or a complete blood count. This is important to test for anemia, but it's also very important because it will help to test for, if there's any white blood cells that tend to be abnormally high, which could be an indication of certain types of infections. So with looking at the red blood cells, you're really looking, is there anemia that's present? What's the hemoglobin concentration? So with the complete blood count, the blood sample, red blood cell characteristics, they'll look at the size, the volume, the concentration of the hemoglobin in the cytosol. They'll also do a platelet count, and they'll also numbers and types of leukocytes. Again, the numbers and types of leukocytes are very important because that, again, if they see one particular type of white blood cell that's elevated, that could be indicative of a particular type of infection. Now, we talked about the process of erythropoiosis, which was the formation of red blood cells. The formation of white blood cells is leukopoiosis, and this is going to be derived from the same stem cells that we saw with the erythrocytes. So leukopoiosis is a process that does occur in the bone marrow, much like the red blood cells, in which those HSCs, the hematopoietic stem cells, form new leukocytes. And it's actually the HSCs will actually be divided into two cell lines, the myeloid cell lines, which will produce most of your formed elements, including erythrocytes and platelets. But then you'll have the lymphoid cell line, which produces lymphoblasts, which will be committed to becoming B cells or T cells. Okay. The myeloid stem cells, okay, will also derive into monoblasts, which will eventually differentiate into mature granulocytes, which then can enter into the bloodstream. B and T cell lymphocytes will mature in different locations. The B stands for bone marrow, and that's where the B cells mature. The T stands for thymus, and that's where the T cells will mature. And you'll notice that in the younger ages, from like toddler up to adolescence, the thymus is very active and it's very large compared to an adult, where the thymus actually does start to atrophy because you're not producing as many mature T cells as you are when you're exploring all these new antigens for the first time. So this just goes down the breakdown of how you make all the different types of white blood cells. Again, the important theme here is that they all derive from the HSCs, and then you have the two main, committed cell lines, the myeloid cell line and the lymphoid cell line. Just be aware of, when you go down the myeloid stem cell line, you're producing your monocytes, basophils, eosinophils, and neutrophils. When you go down the lymphoid cell line, you're producing your T cells and your B cells. Now, some diseases of the white blood cells are called leukemias, and these are typically cancers of blood cells or bone marrow. They're classified as acute or chronic based on the speed of the disease progression. Acute is very rapid increases in immature, non-functional, or poorly functional blood cells. Chronic is a slow accumulation of abnormal, mature leukocytes. The leukemia can also be classified by the cell line from which the abnormal cells derive. Lymphocytic is from lymphoid cell line, generally abnormal B lymphocytes. And myelogenous is from myeloid cell line, and this involves any of the myeloid cells. Acute lymphocytic leukemia, this is a very common leukemia that we find in children. Essentially what happens is abnormal cells crowd the marrow, reducing the ability to manufacture healthy cells, and cancerous cells enter the bloodstream and spread and metastasize to other tissues. The treatment and prognosis for acute lymphocytic leukemia can vary with the type of leukemia. Generally, acute forms metastasize early and require more aggressive management or treatment. The third and final part of our formed elements in blood are platelets. Platelets are small fragments surrounded by the plasma membrane with the following characteristics. They are the smallest of the formed elements involved in hemostasis, which is blood clotting. Platelets do not have nuclei or organelles, and they're actually not even whole cells anymore. They are cell fragments. That's how they're classified. Platelets will contain several types of granules, which contain clotting factors, enzymes, and they may even still contain some mitochondria and glycogen deposits for energy. This will enable them to carry out oxidative catabolism, which is normal breakdown or metabolism that you guys are pretty familiar with. But we'll also get into that when we get to nutrition and metabolism. Platelets will also contain cytoskeletal elements, which include microtubules, which will be associated with actin and myosin filaments. And here's a typical platelet. It's really just a cell fragment that has some of those mitochondria, the microtubules. But again, it really has a lot of those clotting proteins and enzymes that will be very important in the whole hemostasis process. The formation of platelets is by thrombopoiosis. The platelet formation or thrombopoiosis begins again when the HSCs, which are the hematopoietic stem cells, differentiate into committed precursor cells called megakaryoblast from the myeloid stem cell line. Now, what's important is remember all the blood formed elements are coming from the HSCs. Then you have what are called these precursor cells like megakaryoblast for platelets. These are still considered stem cells, but they're what we call committed stem cells, where they will be going down a specific line. A megakaryoblast can't now change its mind to become a lymphocyte. It will become a platelet. The megakaryoblast will develop into megakaryocytes, which will go through repeated cycles of mitosis without cytokinesis. The cell itself never divides, resulting in a massive cell with multiple copies of DNA within a single nucleus. The mature megakaryocytes, when stimulated by a hormone called thrombopoietin, will send the cytoplasmic extension through the clefts in the bone marrow sinusoids into the bloodstream. And what happens at that point is the big megakaryocyte will break off into thousands of little pieces, which are now platelets. Now, platelets actually have a very limited lifespan. They will only last about 7 to 10 days, after which they will be removed from circulation by the liver and the spleen. Again, remember the spleen is also the graveyard for all of your red blood cells. And this is just a breakdown of the formation of platelets there. So here, putting it all together, this is the big picture of all of our formed elements. Again, they all are derived from the hematopoietic stem cell. And you have your red blood cell line, your platelets, and then your various white blood cell lines. All of the formed elements are formed in the bone marrow. Okay. And that is it for all the formed elements and essentially the composition of blood, which includes, the plasma as well as the erythrocytes, the white blood cells, and the platelets that we talked about. So there'll be one more recording for blood, and that will include blood clotting as well as transfusions and blood types. Okay. So there'll be 3 total recordings for the blood chapter. Have a good one, guys.